So I'm just continuing my talk, just highlighting this new way to find drugs. Again, looking for bacteria as the source. So I'm going to my next slide. Okay, you can see here you have a large panel of different type of compounds that can be obtained from bacteria. So you can purify it and use it as anti-cancer drugs. So some of them are antibiotics that kill bacteria, but also are used for cancer cells, but also different type of products. So our interest, in fact, is related with proteins, small water-soluble proteins that come from bacteria, like Ananda Chakrabarty said, one that you have more data and get a peptide from this bacteria and went to the clinical trials phase 1. So also there are another type of proteins. All of them are small proteins that can be, in fact, non-toxic and can be new compounds to trying to treat cancer cells. So you see here this large panel, and I'm going to a little bit further on that. So I'm not going into details about the mode of action of this type of compounds, but of course they can reach the cells by different ways, as you can see here. But there are always problems with drugs, of course, which is the drug resistance. So also in cancer cells just develop resistance to this type of drugs. So you always need to find new compounds, and that's why that we develop it, and you try to go into these small proteins from bacteria that looks like we have a multi-target approach and trying to find out a new compound. So you can see here one of our approaches in Lisbon, in Portugal, to find new compounds. It is based on the genome data that is available right now in the database. As you know, there are many thousands of bacteria which the genome is already published. So doing like a bioinformatic approach, you are trying to find out genes from these genomes that are related with a particular type of scaffold. So you know the scaffold that you are looking for, and this scaffold, of course, you can try to find using bioinformatic tools new genes from these bacteria. So different type of bacteria, you explore, in fact, Pseudomonas aeruginosa as our first bacteria, but then you go for another type of bacteria. And the idea is, in fact, trying to discover new compounds, and then you can isolate the DNA and express and purify using E. coli as a host. And then you can also using a multi-cancer cell lines, trying to see if this type of proteins, in fact, has some cytotoxicity against these cancer cells and not with normal cells like our first protein showed. So this is one of the ways, one of the approaches that we are developing right now, trying to find out new proteins, small ones, water-soluble proteins that you can also and can then go further in this type of studies. Also, you are trying to do, again, looking for bacteria as our source, a new approach. So this is, in fact, a new approach that you are trying to do in our lab. Again, you are not looking in this time to the genomes that are already published and doing some bioinformatic approach in the beginning, but in fact you are doing some experimental work using some type of environmental samples. So you are looking for in Azores, which is an island in Portugal, there are a volcanic part, so there are many type of information about new bacteria that can be isolated from this type of environment. So the idea is to find, and we have samples from these locations, and then extract DNA and do the metagenomics libraries. So the idea is using these metagenomics libraries, using a phage display technology, you can in fact use this phage display to screen again a particular type of target. So we're looking for emerging targets in cancer therapy, and using the alpha screen technology, you're looking for binders to this target. So the idea is to find new proteins from these new bacteria that come from this type of particular environment, and then you can, of course, select the binders that are stronger against these proteins that are particularly important for cancer cells. And then we go for a normal molecular biology technology using and trying to find out these genes, and then you purify the genes, and after that you express and you test it, and you go further in the in vitro studies for further applications. Just a very short overview about our point right now. So I'm put here 4 proteins. So these 4 proteins are in fact the proteins that we are working right now in our lab. So all of them are small proteins. One is the protein that Ananda Chakrabarty in fact found in Chicago, and then you try to find different proteins, like using the bioinformatics approach that I told you before. And all these proteins are in fact studied at different levels. The first one shows in vitro and in vivo data, and also from the proteins, the peptide that have been selected and go to clinical trials, like Chakrabarty said. And but also we have different proteins that are right now studying. One you just find it, so we have no data available in this protein. But another one you already proved it, that this protein shows cytotoxicity against cancer cells and no effects for normal cells. So these proteins are in fact our proteins that we intend to develop. The idea is to go further in the studies. The idea is also, in collaboration with Amrita, start to do some peptides that come from these proteins, because we're talking about a protein. It's a small protein, but of course you need to use an E. coli host to express and to purify. But then if you can select small peptides, we're talking about 20-25 amino acids, you can just chemically synthesize it, and 100% you can use it in our studies, in vitro or in vivo studies. So this is the idea, in collaboration with Amrita, to go further in these studies. So just to finish, so our approach in fact is looking for bacteria, pathogenic bacteria or environmental bacteria that in fact could be new source to find new compounds to treat and to be cytotoxic against cancer cells. So thank you very much.
Thank you.